Hello everyone, this is USABO video slides for part 1 of biochemistry. In this video lesson, we will be teaching about the key concepts for biochemistry in USA Biology Olympiad. Hello, welcome everyone. These slides are the video slides for the USABO Club. Please use these slides as reference to help study and prepare for the exam. Please go over the USABO website for key dates and notes to remember for the exam. We will start this video lesson with biology. Or biochemistry, my bad. So first things first are some preliminary information, which is the atom. The atom is the smallest unit of matter, basically saying everything is made up of atoms, right? Atoms are comprised of three key components, which are found in the diagram on the side, which are neutrons, protons, and electrons. Neutrons have a neutral charge, meaning they have no effect in the actual charge of the atom. Protons have a positive charge, and electrons have a negative charge. The atom's nucleus is the center of the atom. As you can tell, like this area here, that's basically like the cluster of the atom. That's where the nucleus is, and it's only comprised of protons and neutrons, right? Protons are the define the atom themselves, right? So whatever number of protons there is in an atom, that directly correlates to the element itself. So for example, if there's one proton in the element, that directly means that that is a hydrogen atom. If we were to change the number of protons by any means, that would automatically make it a different element and cause different properties of the atoms to occur. So the protons are basically the defining factor. Now let's worry about electron shells. The electrons almost orbit around the nucleus, like the Earth, planets orbit around the sun, right? They orbit around the nucleus, which is P shown in this diagram next above, of the atom. They stay in layers called shells. So the best way I could describe it is that as the planets orbit around the sun, the planets keep in their own safeguarded section around while orbiting the sun. Like the Earth isn't getting any closer to the sun while it's orbiting. It's staying in a relative constant position during orbit. Same with other planets like Jupiter and Saturn. Same thing with the electrons. The electrons have this certain range in which they orbit around the nucleus, and it's a first come first serve basis. So the electrons, they want to be really really close to the nucleus, but basis on how much electrons there is, it could be a little bit more spread apart, and that's why there are these layers, right? The first shell, the blue shell, which is closest to the nucleus, can only hold around 2 electrons, while the second shell can hold around 8 electrons, which is in green, and the third shell holds around 8 as well. We don't need to worry about too much about the third shell, because in biochemistry and in chemistry and in organic chemistry itself, we usually max around the second layer. Let's talk about electronegativity and bonding. Electronegativity is a measure of the atom's ability to pull electrons to itself. So basically, sometimes some atoms are more able to pull other atoms closer to itself, more electrons towards itself, right? This is partially due to the fact that the atom which has a higher electronegativity usually has a higher rate of protons or something, right? These are the basis of many types of bonds, such as covalent and hydrogen bonds. Atoms with high electronegativity are electron grabbers, while atoms with low electronegativity are more electron givers. Let's look at the diagram next to us. I think it's pretty clear to state that fluoride is the more electronegative atom here. Now, it's not defined by the shape, but more of the fact that the electrons are more gravitated towards fluoride than it is to hydrogen here. You can also look at the diagram where it's almost a pear-like shape, where there's abundant mass of color going towards the fluoride side than it is to the hydrogen side, and it's a harder color, representing that electrons are more attracted to it. Now, we don't really need to know the deepest basis on why they're more electronegative, why fluoride is more electronegative than hydrogen in the USABO exam. We just need to have a general idea on how what does this cause, which we'll find out in the next one. But it's really key to understand that high electronegativities likes electrons and want them, and low electronegativity don't like electrons and they'll give them. Let's talk about ionic and covalent bonding, which also result from forms of electronegativity. Now, ionic bonding occurs when one element donates an electron or electrons to another so that both elements can have a full outer shell. Now, here's a really key aspect I want you guys to understand. Electrons want their shells filled. This is partially due to the fact that they want to have, it's something with potential and kinetic energy. We do get a little bit more detail to that in the second part of biochemistry, but in the time frame, let's keep it very simple. We want those shells filled. So they want the first shell to have 2 electrons. They want the second shell to have 8 electrons, so on and so forth. Now, this does not mean that they will take a bunch of electrons just to fill the shell. It's more, in fact, in which scenario is more energy efficient to be able to fill that shell. Adding 7 electrons into their shell or donating just 1, and you have a smaller atom, but you'll have a stable shell. Usually it's the one that's more energy efficient. So let's look at these two in this diagram, the top diagram of lithium and fluoride, right? So lithium has only 3 electrons, while fluoride has 9 electrons. So what does this mean? So automatically both of these electrons have their second shell filled, which is absolutely amazing. But their second shell, that's not quite filled. For example, lithium only has 1 electron in it, and they need 7 electrons to fill it, which is really, really cost effective, right? Really cost inefficient because they need so much energy for those electrons. While fluoride has 7 electron fills, and they need 1 electron. So if lithium could somehow remove its electron and give it to fluoride, that would be fantastic. So it does, and that's what ionic bonding is. Now, the reason why they bond even after they donated the electron is pretty spectacular. Since there is an additional electron in the fluoride and one less electron in the lithium, they almost are like magnets in a sense. Now, here's a really fundamental idea you need to understand. When it comes to atoms, most of the time, if not 99% of the time, the amount of protons is equivalent to the amount of electrons in the atom. So for example, if we have an oxygen atom which has 8 protons, there's probably only 8 electrons in the atom itself as well. So what does this mean? This means that if we ever have an imbalance or shift in the equation for equal amount of protons to equal amount of electrons, that usually indicates that that atom is either more that is positively charged or it's negatively charged. Now in this scenario, since we gave an electron lithium gave an electron to fluoride, lithium becomes positively charged because there's more protons than electrons. Originally there's 3 protons and 3 electrons, but after donating 1, it's 3 protons and 2 electrons, making lithium positively charged. And now in fluoride side, they gain an electron, so it's originally 9 protons and 9 electrons, but now it gained the electron. It's 10 electrons to 9 protons, which makes it an unequal shift, and that makes lithium a positively charged electron and fluoride a negatively charged electron. And like how magnets work, they attract, so they just attract each other. Again, that was a little bit far off detail, but that's just a general idea. Again, this knowledge should pretty much be preliminary, but they're very important because they found so many things. Now, covalent bonding is a much stronger and safer tactic when it comes to sharing when it comes to bonding, where it's just the fact that the electrons share with each other, right? It's just an equal sharing. Well, actually, take that back. It could not be equal sharing at sometimes. It could be a polar covalent bond, in which sometimes one atom is more electronegative than the other atom, like we talked about prior. So what will happen is it will try to mass its electrons towards their side. They're still sharing it, but it's an unequal sharing, almost like when you and your siblings fight over a toy and your mom has favorites, so they probably let them have the toy longer than you do. It's probably that's basically what happens there, right? So when it comes to polar covalent bonds, one has electrons more on their side than the other, right? Electrons are always shared in pairs. I think you can tell by this diagram easily. Like, you don't just do 1 electron shared, it's always 2. Alright, an example is oxygen atoms shown here. And usually if they're the same 2 atoms that are being covalently bonded, there's very little, if not no, unequal sharing. But it's 2 different, like hydrogen and oxygen, wink wink, that might cause an unequal sharing or a distribution. Let's talk about hydrogen bonding. That's what we just talked about, right? It's the attractive force between water molecules that is a dipole interaction, right? So basically what happens is the hydrogen atoms are bound to the highly electronegative oxygen atom, which also possesses two lone pair sets of electrons. Basically, they have two free spots for electrons, right? Making them a very polar bond. The partial positive hydrogen atom of one molecule is then attracted to the oxygen atom of a nearby water molecule. So what does that mean? Well, basically what this is trying to say is that the oxygen is very electronegative, right? So when they bond with the two hydrogen atoms, the oxygen almost hogs the electrons, right? They hog the electrons towards their side, and even though they're not fully taking in the electron, it's almost as if that side has an imbalance, like we talked about with the ionic bonding, right? It's almost as if there's a more radical shift in electrons than there is protons, causing it to be negatively charged, and the hydrogens not having electrons as much become positively charged. So they're partially positively charged, which is the hydrogens, and they're partially negatively charged, which is for the oxygen. And that partiality also then works with other water molecules and causes that hydrogen bond. It's a really easy topic when you think about it. I really recommend watching videos like Khan Academy and other resources to get a deeper grasp in hydrogen bonding, because it's a very fundamental concept. Let's now talk about the properties of water deriving from hydrogen bonding. So when water molecules connect with other water molecules, just like in this let me just go back a little bit. In this diagram right here, that's called cohesion. When they work with other polar molecules or ionic molecules, that's called adhesion. And a really great example is actually the xylem of plant cells, right? Let's look at the xylem right here. As you can tell that in the plant cells themselves, right, the water is what do you call having cohesion by hydrogen bonding with each other, but then they're also hydrogen bonding with the polar stems of the xylem, and this helps it go against gravity, and pull water upwards through the plant instead of it going down. If without this systematic, it would actually be impossible for plants to grow. There's another property of water called surface tension. Since water molecules are not hydrogen bonding to each other, but not the outside environment. Sorry for the misspelling here. Basically, what it's trying to state here is the water is very peculiar in a sense, right? It likes to be with its own kind. So what happens is when it's faced with the outside environment, such as air, such as air, or maybe an oil, right? They create a film around themselves as a barrier made of hydrogen bonds against the outside environment, shown here, right? You can see where it says air here, and there's those 5 yellow balls that represents water molecules, and that's how they almost make a film to protect the other water molecules from the outside environment. It's all, and if you break this film, right, it causes like, it causes a bunch of things. Like, it causes the ability to like mix solutions into it, right? The water molecules almost make a film around the water to the outside environment to make it harder to break the barrier, right? And then water striders, which is the organism found in the bottom, they basically don't break it because they're so light. You don't really need to know that. I just found it cool to say. Next is specific heat. Since water has hydrogen bonds, it requires more energy to break those bonds. Already they're covalently bonded, and now there's an additional layer of hydrogen bonds to break, right? If you want to heat up water into gas, right, you have to break those bonds to be able to form it to gas. So this is another layer of bonds that you must work with to break it, right? So that's what we're talking about. It needs a lot of energy to break the bonds and to be able to disperse the molecules, right? That form of energy to the form of energy to break the water molecules is heat, right? So the system to break the water molecule and the hydrogen bonds is heat themselves, aka thermal energy, right? Since it needs a lot more energy compared to other molecules to break the bonds, water has a high specific heat. So what does it mean, right? Let's say you put a pot of water at the in your stove, and you put it at the highest heat. Within like 5 to 10 minutes, that stove is frantically hot. Like, you touch it, you're gonna burn your finger. But if you touch the water of it, it's sort of lukewarm, right? It's not that hot to touch it within the beginning of it. That's because of its high specific heat. It requires so much energy to break the bond. It takes a while for it to occur. So that when you touch it, it's still lukewarm. It takes a while for the bonds to break. And that's why it's specific heat. Now, the great thing is the high specific heat of water allows it to make it harder to evaporate, which allows oceans to stay cooler for longer, and a bunch of other properties for life. Now, temperature is a bifactor of thermal energy, and thermal energy is a form of kinetic energy. So temperature is just a measure of that kinetic energy, aka thermal energy. The next concept is evaporative cooling. Right? So basically, when we break bonds, energy is released, right? This is a very key concept that you're going to learn in Gibbs free energy, which is going to be the next video. But basically what happens is when we break bonds, energy is released, right? So when energy is released, let's say we are boiling water. We are boiling water, so we're breaking the hydrogen bonds and we're breaking the bond between the molecules of oxygen and hydrogen itself. So that produces a lot of heat released, okay? And that's what causes evaporative cooling, right? Let's say you're playing a really good game of basketball, right? You've been running and jogging all day, right? What happens is you start sweating, right? That sweat actually, when you sweat, does it make you feel hotter or do you feel cooled down? You feel cooled down because of evaporative cooling. Basically, what's happening is the bonds are breaking, and so much thermal energy, and thermal heat is being released out of you. You're cooling yourself down. Taking heat away from a source cools it down. So that's basically what's happening. And the diagram that we show here is basically what happens. We apply ice packs all over the body if the patient's really in a high temperature, because not only are they sweating trying to cool themselves down, but when they're sick, it's much harder to break those bonds. So that's why ice packs are added to actually help aid them to break the bonds and cool themselves down. This diagram is what I just found off the internet, but I just want you to understand evaporative cooling. Alright, next one is a very important property about ice. So ice is less dense than liquid water. So what does that mean? Well, since ice is less dense than water, when the temperature drops, instead of the water just like freezing as it is, the water molecules rearrange into a lattice-like pattern shown in the diagram next to us, causing many gaps of space in the setup, making it less dense. It's a high surface area with many holes, that's basically making it less dense, right? And because of that, it allows to be able to float in liquid water. And because of that, it can act as a protective barrier in very cold temperatures, allowing the liquid water to stay liquid, while the ice protects from the harsh weather, allowing life to continue. So that's really what the ice density does. Let's now talk about biological macromolecules. There are four main classes of macromolecules. Think of macromolecules like Lego, being the same/similar pieces that are repetitively used to make complex structures, right? The building blocks of the Lego brick is called a monomer, and they're connected by covalent bonding. Now, the actual end piece of Lego set finished product is called a polymer. Now, there's four main types of macromolecules, which are carbohydrates, lipids, proteins, and nucleic acids. Let's first start with carbohydrates. Carbohydrates are sugars and starches. So basically, sugar, cornstarch, potatoes, bread, wheat, those are all carbohydrates. The monomer is called a monosaccharide. So the building block, that's Lego piece of a carbohydrate, is called a monosaccharide. The Lego end product, the building or whatever you built, right, is called a polysaccharide. And two monomers are called disaccharides. That's not really important unless you want to talk about like specifics like fructose and lactose, but that's not the point. Now, that's just the names for carbohydrates. That's basically what you need to know for carbohydrates. But now let's talk about a very key aspect when it comes to these macromolecules on how they are bonded. They're bonded by something called dehydration, synthesis, right? So basically what happens is two monomers bond together by removing water. So when they bond, right, they take water as a byproduct, and that allows them to bond. And that's called dehydration synthesis. We're dehydrating or taking water from the monomers to itself to help them bond. That's shown in the diagram right here, right? You can see how water is being extracted after they bond, and then to break those bonds, this is the complete opposite. We just add water to it, and the bonds are automatically broken. Adding water molecule to split the monomers, right? Alright, our next main topic is called lipids, okay? These aren't true macromolecules because they don't fit the designated category of a macromolecule, but they're so abundant and so versatile and important that we plug them into the macromolecule system, right? The reason why they can't do that is because they don't have a definite monomer, and they can't be formed into long polymer chains. They're a diverse group of macromolecules, one of, if not the most diverse, and one common factor of lipids is they all share a hydrophobic region, or a region in which they hate or loathe water, and also emphatic region, the region right there, where they have sections that like water and sections that they don't like water. A very prime example is about to come up. Now, the three main types of lipids that we need to worry about are triglycerides, phospholipids, and steroids. Sorry that the image is rotated. I just wanted a bigger picture so you guys could understand. We're now going go to the next slide and explain these three. So triglyceride, right? Triglyceride. We're just gonna call it triglyceride. It's a key type of fat under triglycerides, right? And it's made of one molecule of glycerol, so that blue part in this diagram right here, the top diagram, and one molecule as a backbone, and three fatty acid tails. So those three lines coming out of it, right? Fatty acid tails are nonpolar due to these small differences in electronegativity between the hydrogen carbon, preventing interaction with water molecules and lipids, and making lipids hydrophobic. So basically, the reason why lipids don't like water, or like fats don't like water, is because they are not nonpolar. They're equally sharing, and because they equally share, they don't have any designated need to be any. They don't like to be with molecules that don't equally share their electrons, or they don't share at all, like ionic. So that's why they're hydrophobic, right? They don't like molecules that don't share their electrons. To keep it as simple, right? So let's talk about two types of fats, which are saturated fats, contain no bend in fatty acid chain, solid in room temperature, and then unsaturated fats contains kinks in fatty acid chain, liquid in room temperature. So the heck does that mean? Let's look at the two diagrams in the yellow box here. The top one is called a saturated fat chain, right? So basically, you can see how the chain is in a perfectly straight line. That basically means there's no bend in the fatty acid chain, in the fatty acid chain, and that when it's stacked continuously on top of each other, it's actually solid in room temperature. So examples can be like ghee, coconut oil, lard, and butter. These are very prime examples. And then unsaturated fats contain kinks. So let's look at the second diagram in the bottom. You can see how that red portion has a double bond of carbon. It almost causes a bend in their shape, right? And now when you stack those together, it's not as tightly packed, causing it to be liquid in room temperature. So olive oil, canola oil, peanut oil, they all are having a kink in their bends, right? Yeah, that's basically it. Phospholipids. This is where we talk about the prime example for emphatic molecules, right? Phospholipids are emphatic, having regions of hydrophobic and hydrophilic. So they're composed of glycerol, that red circle in the top diagram, two fatty acid tails, the two yellow parts, and a phosphate head, the smaller inner yellow circle in the red circle. When phospholipids are introduced into a water solution, they form a phospholipid bilayer. So what happens, right? Since the head loves water, they are facing towards the water environment. And since the tails hate water, they're facing towards an environment that are outside of the water. So when they form a phospholipid bilayer in which the tails face each other and the heads face the water. So that's basically it. And this is also the basis for our plasma membrane, which we'll get into detail very, very soon in the next video, actually. Now, steroids, they're the third group of molecules classified under lipids, and their only basis and their only point of recognition is that they have 4 rings. So you can tell by these two diagrams that they have 4 rings. Right? And these 4 rings are found in every single phospholipid, or every single steroid, and what makes them unique, what makes each steroid unique, is what's added onto them. Let's now talk about proteins. This is probably the most important macromolecule and the one that we're going to go the most in depth to, right? Monomers of proteins are called amino acids. They're composed of a carboxylic acid group, amine group, and an R group. So let's look at the top diagram here. You can see how there's a blue part, which is the amine group. There's a red part called the carboxylic acid group, and there's an R group, which is a variable group. Let's talk about that R group real quick. R groups is like the X variable in math, being a placeholder. Like when we do math equations, X does not have a definitive value. They could be anything depending on the equation or the question itself, right? So that R is a placeholder and can represent 20 unique molecular structures that can be added into the protein, all right? And that causes special functions of it. Amino acids are bonded by dehydrogen synthesis, as they told before, and their polymers are called polypeptides/proteins. And protein folding is what gives proteins a more unique structure and function, and we'll go into very much detail on this in the next video. Our next topic is nucleic acids, right? Nucleic acid is a basis of our genetic code. Their monomer is called nucleotides, and they're composed of pentose, nitrogenous base, and a phosphate group. The basis of the nucleic acid is as 2 types: pyrimidines, which are 6-atom ring of carbon and nitrogen. Examples of the bases are cytosine, thymine, and uracil, while pyrimidines are 6-atom ring with 5-atom ring fused together, made of carbon and nitrogen, which are adenine and guanine. polymer is called a polynucleotide or nucleic acid, and the DNA and slash RNA is joined by the sugar phosphate backbone. So this yellow and red part in the diagrams here, right? That's the backbone of it. That's what holds that, what do you call it, macromolecule together. The covalent bonds form the creation of the sugar phosphate backbone are called the phosphodiester bonds. Basically, the way it works is the phosphate bonds to a sugar and repeat. So those bonds are called phosphodiester bonds. In DNA, the sugar in the nucleotide is called deoxyribose. That red part in the diagram right next to it is called deoxyribose, and the DNA is in double helix shape, so it looks like. I don't know how to describe a double helix. It's a pretty easy shape to understand, right? It's like just two spiraling lines, probably. They're connected by base pairing, right? Which is the adenine to guanine, the cytosine to thymine, the adenine to uracil, depending if it's RNA. That's how they're bonded by. And the strands are anti-parallel. So basically, one strand goes one way and the other strand goes the opposite direction. So one goes north, one goes south, just like a highway. And we describe it as one going from 5 carbon to 3 carbon, and another strand going from 3 carbon to 5 carbon. You don't need to worry about that too much, though. All right, nucleic acids. The format of the way that we are built is because of proteins. Right? Everything that we do, everything that we're composed of, is because of proteins. But how are proteins formed? It's performed by a very simple process. DNA is created. DNA is then transcribed to RNA. RNA acts as instructions for amino acids, like Lego booklet instructions, and then that amino acid is made and it's made into protein synthesis. Alright, so it's called the central dogma, and this system allows for all proteins and all types to be made. It's effortless and efficient. We'll go more into detail into this in the genetics unit, right? So now each amino acid is represented by one or more codon, which are triplets of RNA or DNA nucleotides. Basically, just 3 random variables of nucleotides. So GGC, ACT, TTA, CCCC, anything like that, right? And these 3 codons represent an amino acid, right? So GGC in a special chart, which is an amino acid chart, would represent a specific type of amino acid, and that's how the proteins read it, and they'll create that amino acid. Okay, so that's basically it for biochemistry part 1. If you have any questions, please feel free to contact me. And the newer videos will be out very, very soon. I hope you guys enjoyed this. If you have any questions, because this is a lot of info to take in, don't hesitate to ask me. And I hope you all have a great day. Thank you.